At a depth of 32 meters below the surface of the Pacific Ocean, where sunlight arrived only as a trembling blue-green glow, a young giant Pacific octopus pushed one arm out from beneath a shelf of volcanic rock. The arm moved slowly, almost thoughtfully. Hundreds of suckers touched the stone one after another, tasting the water, feeling tiny changes in pressure, and reading the chemical traces left by passing animals. To a human observer, the movement might have looked simple. In reality, every centimeter of that arm was processing information. The octopus had no name in the way humans understood names. Later, a marine biologist would call her Nera, but for now she knew the world through touch, smell, light, texture, and motion. Her body was soft except for the hard beak hidden at the center of her arms. She had no bones and no external shell. That softness allowed her to squeeze into spaces that seemed impossibly small, provided her beak could fit through the opening. On this morning, she was hiding in a den whose entrance was barely wider than a diver's wrist. Nera had 3 hearts. 2 branchial hearts pumped blood toward her gills, while a larger systemic heart pushed oxygenated blood through the rest of her body. Her blood was not red. It was blue because it used hemocyanin, a copper-containing protein, to carry oxygen. Hemocyanin worked well in the cold, oxygen-poor water where she lived, though it was less efficient than the iron-based hemoglobin found in humans. For that reason, Nera rarely wasted energy. She preferred crawling over rocks to long periods of jet propulsion. Still, she was a hunter. A crab moved across the seafloor several meters away. Nera saw it with eyes that, despite having evolved separately from vertebrate eyes, worked in a surprisingly camera-like way. She could judge brightness, contrast, polarization, and movement. Her skin changed before she even moved. Dark brown patches spread across her mantle. Pale streaks appeared along her arms. Small muscular structures in the skin raised bumps called papillae, making her smooth body resemble rough stone covered in algae. The change was not merely color. Her skin contained chromatophores, tiny elastic pigment sacs controlled by muscles and nerves. By expanding and contracting them, she could display yellow, orange, red, brown, and black tones. Beneath, these were iridophores and leucophores, layers that reflected and scattered light. Together, these structures allowed the octopus to alter its appearance in fractions of a second. The crab did not react. Nera flowed forward. Her 8 arms did not move like the legs of a land animal. Each arm contained dense networks of neurons. A large portion of an octopus's nervous system lies outside the central brain, especially in the arms. This does not mean the arms are independent animals, but they can perform complex local processing. A sucker can touch, grip, and detect chemicals while the central brain coordinates the broader goal. Nera's front arms reached the crab first. The crab raised its claws, but the suckers attached to its shell with sudden force. The octopus folded around it like a living net. Hidden between the bases of her arms, her beak closed on the crab's body. Salivary secretions containing toxins and digestive enzymes began to weaken the prey. Within minutes, the struggle ended. She carried the crab back to her den. Outside the entrance lay a small pile of shells. Scallops, broken crabs, and fragments of clamshells formed what marine researchers called a midden. To another octopus, or to an experienced diver, the pile was a sign that the den was occupied. Several hundred meters away, a research vessel drifted above the reef. Dr. Mira's son leaned over a monitor inside the vessel's compact laboratory. She had spent 12 years studying cephalopod behavior. Squid, cuttlefish, nautiluses, and octopuses all belong to the group Cephalopoda, but octopuses fascinated her most because they combined a radically different body plan with remarkable behavioral flexibility. On the screen, a remotely operated vehicle moved slowly over the seafloor. Its lights crossed sponges, sea stars, anemones, and patches of cold water coral. "Stop," Mira said. The pilot froze the vehicle. "There." Shell pile. The camera turned. A dark shape shifted beneath a rock. Mira smiled. "We found one." The team did not capture the animal. Instead, they placed a small observational camera several meters from the den. Its lights were dim and filtered to reduce disturbance. They also positioned a puzzle box nearby. Inside the transparent box was a piece of crab. The lid could be opened by rotating a simple latch. The experiment was designed to study exploratory behavior. For the first 6 hours, Nara ignored it. That did not disappoint Mira. Wild octopuses were cautious. Curiosity and fear could exist at the same time. An unfamiliar object might contain food, but might also conceal a predator. By the second night, Nara approached. One arm touched the box, then another. Her suckers moved across the acrylic surface, sampling traces of dissolved chemicals. She could smell the crab, but could not reach it. She pulled at one corner. Nothing happened. She pushed the box. It rolled slightly. For 20 minutes, she explored almost every surface except the latch. Then she left. The next evening, she returned and went directly to the lid. Mira watched the recording in the morning. "Look at that," she whispered. Nara pulled once. The lid held. She changed her grip, placed 2 arms against the side of the box, and rotated the latch with a third. The lid opened. She took the crab. Mira replayed the sequence several times. She was careful not to call the behavior human-like intelligence. Intelligence had evolved under many different pressures, and comparing every animal to humans could hide what made its mind unique. An octopus did not need language, hands, or a long childhood to solve problems. Its intelligence had developed in a body with 8 flexible arms, no skeleton, strong predatory instincts, and relatively little social life. That difference was scientifically important. The common ancestors of humans and octopuses had lived more than 500 million years ago. Since then, vertebrate and cephalopod nervous systems have followed dramatically different evolutionary paths. Yet both lineages produced large brains, sophisticated senses, memory, learning, and flexible behavior. To Mira, Nara was not evidence that octopuses were almost human. She was evidence that complex cognition could be built in another way. Over the next several weeks, the researchers observed Nara without touching her. They recorded hunting, grooming, den maintenance, camouflage, and sleep-like behavior. At times, her skin changed pattern while she rested, flashing through colors and textures, even though nothing obvious in the environment had changed. Researchers studying octopus sleep had found periods resembling quiet sleep and more active states in which rapid changes in skin pattern occurred. Whether these episodes represented anything like human dreaming remained uncertain. Mira refused to call them dreams, not because the possibility was impossible, but because science required evidence. One night, the camera recorded something unexpected. A moray eel entered the edge of the frame. Nara saw it immediately. Her body became pale, then dark. She flattened against the rock and pulled her arms close. Moray eels were dangerous predators. An octopus could escape by jetting water through its siphon, but rapid jet propulsion consumed considerable energy and reduced the efficiency of the systemic heart during intense movement. The eel came closer. Nira waited. At less than a meter, she exploded from the rock. Water shot from her mantle cavity through the siphon, propelling her backward. At the same moment, a cloud of ink spread through the water. Octopus ink was more than a dark visual screen. Its major pigment was melanin, mixed with mucus and other compounds. Depending on how it was released, ink could form a diffuse cloud or a denser blob that might confuse a predator. Some components could also interfere with chemosensory tracking in certain attackers. The eel snapped at the black cloud. Nara vanished between two boulders. For 4 days, she did not return to the den. Mira worried that the animal had been injured or killed. Predation was ordinary in the ocean, but months of observation had made Nara scientifically valuable. More than that, Mira had begun to feel the strange attachment researchers often developed toward individual wild animals they studied closely. On the 5th night, Nara reappeared. One arm was shorter. The camera showed a ragged, healing end where part of the arm had been lost. Mira enlarged the image. She escaped by a few centimeters, she said. Octopuses can regenerate damaged arms. The process is not instantaneous. New tissue must form, nerves must reconnect, muscles must develop, and suckers must gradually reappear. Regeneration can take weeks or months depending on the species, injury, temperature, and health of the animal. Nara seemed to compensate immediately. She hunted with 7 fully functional arms and used the injured one less often. The team marked the date and began measuring the arm's recovery from the video. After 12 days, a small taper extension was visible. After 29 days, the new section moved more freely. After 7 weeks, tiny developing suckers could be seen. Mira considered this one of the most beautiful results of the project. Regeneration was not merely repair. It required tissues to rebuild a complicated structure that normally contained muscles arranged in 3 dimensions: sensory receptors, skin, blood vessels, and an extensive nerve cord. Nara's body was solving an engineering problem no laboratory could yet reproduce with comparable elegance. During the same period, the researchers noticed that her hunting strategy changed with different prey. With crabs, she often pounced and enclosed them. With clams, she sometimes pulled the shell apart using suction and muscular force. When a shell resisted, she drilled a small hole using structures in her mouth and introduced secretions that helped weaken the prey inside. She was not using one fixed method. She selected among techniques. To test memory, without training her in captivity, Myra's team introduced two containers. One had a rough surface and contained food. The other was smooth and empty. Their positions were changed each night. Nara quickly began approaching the rough container first. After several days, the food was removed. She continued choosing the rough container for 3 nights before gradually reducing her interest. The results suggested associative learning. A sensory feature had become linked with a reward. Mira wrote in her notes, "Flexible learning is especially important for animals living in variable environments. An octopus encounters prey with different defenses, changing shelters, predators, currents, and unfamiliar objects. A nervous system that can update behavior from experience may provide a strong survival advantage." Months passed. The seasons shifted. Nara grew. An octopus grows rapidly because most species have relatively short lifespans compared with many large vertebrates. Giant Pacific octopuses may live several years, longer than many smaller octopus species, but even their lives are brief compared with whales, turtles, or humans. The brevity seemed strange to Mira. A large nervous system was metabolically expensive. Learning required time. Yet evolution had produced a highly capable animal that often lived only a few years. The answer was not one single factor. Cephalopod evolution had followed its own reproductive strategy, metabolism, ecology, and life history. Many octopuses grow quickly, reproduce once, and die after breeding. This pattern is called similarity. One afternoon, the team found evidence that Nara's life was entering a new phase. A second octopus appeared near the den. It was male. Male octopuses possess a specialized arm, called the hectocotylus, used to transfer spermatophores containing sperm to the female. Courtship can be cautious and dangerous. Octopuses are usually solitary predators, and a meeting between adults can become aggressive. The male remained at a distance. His skin shifted through contrasting patterns. Nara moved partially out of the den. For nearly an hour, the two animals approached and withdrew. Then the male extended the specialized arm. The mating process was partly hidden by rocks, but the researchers captured enough footage to identify it. Afterward, the male disappeared into deeper water. Mira knew what might come next. Female octopuses can store sperm for some time before fertilizing their eggs. When conditions are suitable, a female selects a protected den and lays thousands, sometimes tens of thousands, of eggs depending on the species. Weeks later, Nara stopped using the exposed hunting den. The tracking cameras found her in a deeper crevice with only one narrow entrance. When the researchers finally positioned the camera, Mira felt both excitement and sadness. The ceiling of the den was covered in strings of small white eggs. Nara had become a mother. From that day, her behavior changed dramatically. She rarely left the eggs. She cleaned them with her suckers, removed debris, and directed fresh oxygenated water over them using her siphon. Flow was essential. Developing embryos needed oxygen, and stagnant water could allow waste or microorganisms to accumulate. Nara's movements became repetitive and careful. She guarded the entrance. A crab walked within reach. She did not hunt it. Days became weeks. Her body slowly lost mass. In many octopus species, brooding females eat little or stop feeding entirely. Hormonal changes associated with reproduction trigger major changes in behavior and physiology. Research on octopus reproductive biology has shown that structures such as the optic glands play a central role in the transition toward brooding, decline, and death. Mira had explained similarity to students many times. Watching it happen was different. The animal that had opened puzzle boxes, escaped an eel, regenerated an arm, and adapted its hunting strategies was now spending nearly all of her remaining energy protecting embryos she would never raise. The eggs developed. At first they looked like pale droplets. Later, dark points appeared inside them. Eyes. Under magnification, the embryos could sometimes be seen rotating within the egg capsules. Their chromatophores began to function before hatching, producing tiny flashes of pigment. Mira watched the monitor for long periods. "You can see the nervous system coming online," she told a graduate student. That phrase was metaphorical, but the developmental process was real. Neurons differentiated. Sensory organs matured. Muscles became coordinated. The small bodies prepared for a world they had never experienced. Nera continued to weaken. Her skin appeared loose. Small lesions developed. She spent less time rearranging rocks around the den and more time resting beneath the eggs. The researchers debated whether to intervene. They could have offered food directly. They could have tried to remove eggs to an artificial incubator if the female died early. But the project had been designed around minimal interference. More importantly, Nera's decline was not an accidental emergency. It was part of the natural reproductive cycle of her species. Mira made the decision. They would observe. They would not turn a wild animal's life into a laboratory rescue simply because its natural ending made them uncomfortable. The choice was scientifically defensible. It was still painful. One evening, a storm crossed the surface. 30 meters below, the reef did not experience crashing waves, but currents intensified. Fine sediment moved through the water. Nera positioned herself beneath the egg strings and pulsed water across them again and again. For 9 hours, she barely rested. By morning, the current weakened. The eggs remained attached. Several days later, the first one hatched. A tiny octopus emerged, almost transparent. Then another. Then dozens. The hatchlings were miniature in structure, but not simply miniature adults in behavior. Depending on species, young octopuses may enter a planktonic stage, drifting and hunting small organisms in the water column before eventually settling to the bottom. Nera's hatchlings rose from the den like living sparks. Some changed color. Some spread their arms. Some disappeared immediately into the dark water beyond the camera lights. Thousands of evolutionary instructions, encoded in DNA and shaped by development, were now being tested against the ocean. Most would not survive. Predators would take many. Currents would carry others into unsuitable habitats. Food shortages, disease, and chance would eliminate more. But a few might reach adulthood. Natural selection did not guarantee survival to the strongest, smartest, or most beautiful individual. It acted through differences in reproductive success across generations. Traits that improved survival or reproduction under particular environmental conditions could become more common, provided those traits had heritable components. Nera had already done what evolution required. She had reproduced. For 2 more days, eggs continued to hatch. Then the den grew quiet. Nera stopped ventilating the remaining egg strings. Her arms curled loosely beneath her. The systemic heart that had pumped blue blood through her body for years slowed and finally stopped. Mira watched the final recording without speaking. No dramatic event marked the moment. 1 second the octopus moved, then she did not. The graduate student beside Mira said, "It feels wrong that an animal that intelligent gets such a short life." Mira looked at the screen. Short according to whose measurement? The student said nothing. Her life wasn't unfinished, Mira continued. It was shaped differently. They recovered the camera several days later, but left the den untouched. Back in the laboratory, the team began analyzing months of data. Nera's movements became coordinates. Her color changes became coded behavioral categories. Her puzzle interactions became latency measurements. Her arm regeneration became growth curves. Her choices became statistics. Science required reduction. A complex event had to become something measurable if researchers wanted to compare it, test it, and learn from it. But Mira worried about reduction when it became forgetting. A graph showing time to open container did not show the slow sweep of Nera's suckers across the lid. A data point labeled predator escape did not show the instant when her body changed from rock-like camouflage to explosive motion. A line showing arm length over time did not show how she adapted while injured. Numbers were not the opposite of the story. They were another way of preserving parts of it. A year passed. The research vessel returned to the same reef. Mira joined the expedition, though the original project was complete. She told herself she wanted environmental measurements for comparison. Temperature, oxygen, species abundance, current speed. That was true. It was not the whole truth. The remotely operated vehicle descended through darkening water. At 30 meters, familiar rocks appeared. The old den was empty. The midden had scattered. A sea star crawled over the place where Nera had once hidden. The vehicle moved on. 10 minutes later, the pilot said, "Possible octopus." 2 o'clock. Mira looked at the screen. A small octopus rested beside a sponge. It was much younger than Nera had been when they first found her. As the camera approached, the animal changed from mottled gray to dark brown. One arm lifted. Its suckers touched the water and nearby stone. The octopus did not flee. Mira knew there was no way to determine from appearance whether this individual was one of Nera's offspring. Thousands of hatchlings had dispersed. Ocean currents could move juveniles over great distances. Genetic sampling might estimate relatedness, but no sample would be taken simply to satisfy sentiment. The animal crawled toward a narrow crack. Its body compressed. The mantle followed the arms. In seconds, the octopus disappeared through an opening that looked too small to contain it. The pilot laughed. "Never gets old." Mira watched the empty crack. "No," she said, "it doesn't." That night, she stood on the deck beneath a sky without city lights. The ocean around the ship looked black, but she knew it was crowded with activity. Below the surface, animals exchanged gases, hunted, hid, reproduced, migrated, competed, cooperated, sensed, learned, and died. Every organism carried a history written partly in genes and partly in experience. The octopus represented one of evolution's most extraordinary experiments. Its ancestors had once possessed more obvious shells. Over evolutionary time, most octopus lineages lost the external shell that protected other mollusks. The loss made their bodies vulnerable, but it also made them flexible. Without armor, they evolved other defenses: camouflage, dens, venom, speed, ink, intelligence, and extraordinary control of the skin. Vulnerability and possibility had arrived together. Mira returned to the laboratory with a short video of the young octopus. She added it to the archive, but did not include it in a publication. There was no experiment attached to it, no statistical result, no claim. Still, she watched the clips sometimes. The young animal changed texture, extended an arm, sampled the rock, and vanished. A complete sequence lasted 11 seconds. In those 11 seconds were millions of years of evolution. The muscles of the arm were built from genetic instructions inherited through generations. The suckers performed sensory tasks refined by selection. The chromatophores opened under neural control. The soft body compressed into a crevice because a lineage that had lost its shell had gained a new kind of freedom. No single octopus understood that history. Neither did Nera. She had never known the words neuron, hemocyanin, chromatophore, evolution, regeneration, or simulparity. She had never needed them. Science belonged to the observers trying to explain the mechanisms. Life belonged to the animal living them. Years later, Mira wrote a book about cephalopod biology. She described laboratory research, field studies, comparative neuroscience, conservation, and unanswered questions. She wrote about ocean warming, acidification, deoxygenation, habitat alteration, pollution, and fishing pressure. Some octopus populations might adapt to changing conditions. Others could decline. Their responses would depend on species, location, prey availability, reproductive biology, and the speed of environmental change. She resisted simple predictions. The ocean was too complicated for slogans. But she ended the final chapter with the reef. She described the first arm emerging from Nera's den. She described the puzzle box, the moray eel, the regenerating tissue, the white strings of eggs, the hatchlings entering dark water. Then she wrote about what the octopus had taught her. Not that animals were secretly people. Not that intelligence followed one ladder from simple to advanced. Not that evolution had a destination. The lesson was almost the opposite. Evolution branches. Bodies create possibilities and constraints. Nervous systems solve problems in forms shaped by those bodies. An octopus experiences the world through a sensory and motor system radically different from ours. Yet it can learn, remember, explore, choose, escape, manipulate, and adapt. That combination should make humans cautious about treating unfamiliar minds as lesser minds. One morning, many years after Nera's death, Mira visited an aquarium before opening hours. A keeper led her to a large tank containing another giant Pacific octopus. The animal was awake. Mira stood near the glass. The octopus approached. Its eye turned toward her. One arm spread across the transparent wall. suckers attached to the other side of the glass, only centimeters from Myra's hand. She placed her palm against it. There was no direct touch, no shared language, no reason to imagine the animal understood who she was. Still, two nervous systems, shaped by more than 500 million years of separate evolution, were briefly oriented toward each other. Mira looked into the octopus's eye. She thought of all the questions that remained. How does an octopus integrate information from 8 arms? How are memories represented in its nervous system? How much local autonomy does an arm possess during complex behavior? What is the relationship between camouflage control and visual processing? What subjective states accompany exploration, rest, injury, hunting, or sleep? How do different octopus species vary in cognition? How will rapidly changing oceans affect their development and behavior? Each answer would produce more questions. That was not failure. That was the structure of science. The octopus withdrew its arm. Its skin brightened. Then dark waves passed across the mantle, and the animal turned toward a pile of rocks at the back of the tank. Within seconds, it seemed to dissolve into the background. Mira could still see the outline because she knew where to look. Then even the outline became difficult to find. She smiled. An octopus could hide from the eye, but not from curiosity. And curiosity, in both the animal and the scientist, was where the next discovery began.